The first step of gene expression is called replication. This is when the top strand of DNA is copied. During this stage, the double helix goes through the helicase, which is an enzyme that separates the two strands. The top strands goes through the 5 and 3 prime part of the DNA polymerase, while the bottom strand goes through the 3 and 5 prime parts of the DNA polymerase. While in the DNA polymerase, new DNA base pairs get attached to the top strand that is going through it. The DNA polymerase continues to go down the entire strand and reading the codons on the DNA. After it leaves the DNA polymerase, the newly formed helix detaches again and goes into the RNA polymerase, which is an enzyme that carries the genetic instructions from the DNA. The 5 to 3 prime strand goes through the top of the RNA polymerase, and the 3 to 5 prime strand, the newly made DNA, goes through the bottom of the RNA polymerase. When this happens, RNA is being produced. When the RNA is made, it goes through the active site. After going through the RNA polymerase, the two strands that were separated attach back to each other. After going through the RNA polymerase, the strand goes into the ribosome. The ribosome is where the mRNA gets translated into protein. tRNA, which brings amino acids into the ribosome and contains the anticodons for the mRNA strand, then enters the ribosome in the P site and continues to read the codons until a match enters into its site and it attaches to the RNA strand. Attached to the top of the tRNA is the methionine amino acid. Another tRNA enters through the A site, and the tRNA that was in the P site goes into the E site, while the amino acid that was attached to it leaves through the protein exit channel. As more amino acids leave They attach to each other. Then the stop codon attaches to its anticodon on the mRNA strand. This, all the amino acids that were attached to each other form a structure and go on to do their function in the body.